So tonight we're going over Chapter 15, and in Chapter 15 it introduces some of the molecular biology techniques that are used in biomedical research. So this is used to research diseases and discover new treatments, and also in both industry and forensics. So I just wanted to let you know I'm going to be gone for the next couple weeks out of town, and so this last section of the course, the lectures are going to be only the archive. And I'll post it online too, but it's if you go on the archives, so if you go on the BIOL and click the link to the archives and do a search for my last name, it'll come up with last semesters, and starting with November 14th through December. That's all the lectures for the last portion of the semester. So like I said, I'll post the specifics online, but I just wanted to let you know, especially those of you that have been coming live to the lecture. So 99% of human DNA has exactly the same sequence between individuals, but single nucleotide polymorphisms exist. So these are a variation in a single base pair that's present in over 1% of the population. So different alleles of most genes differ by only one base pair, and so variation in alleles is the basis for the variation in human traits. And this is what makes each person unique. So these single nucleotide polymorphisms, or SNPs, or SNPs, account for many of the differences in how likely someone will develop a disease such as cancer or Alzheimer's, also whether the person will have a good outcome from various drugs and therapies. So therefore, personal genetic testing may soon revolutionize medicine. And this is because it would allow physicians to customize treatments on the basis of an individual's genetic makeup. So the chip shown in the slide can show which base pair you have for over 9,000 SNPs, or locations of these single nucleotide polymorphisms that occur in a person's DNA. So unfortunately, at this point, though, scientists and doctors don't actually know how to use this information to get better treatment yet. So, but this is an area that in about 5 to 10 years, it'll be cheap enough for everyone to use the technology, and so therefore the research will be much further along, and hopefully physicians will be able to personalize treatments and understand the risk for various genetic diseases. One of the most important discoveries made after learning the structure of DNA was restriction enzymes. So unfortunately, when the structure of DNA was first discovered, studying stretches of millions of base pairs in each chromosome was completely impossible. So then it was noticed that certain bacteria are resistant to infection by viruses. So the resistance that these bacterial organisms had comes from the fact that they produce enzymes that specifically chop the viral DNA before it can be integrated into the bacterial genome. And these enzymes are a defense mechanism against the viruses, and they restrict the viral growth. This is why they're called restriction enzymes. So restriction enzymes recognize specific sequences in the DNA and only cut at that sequence in a predictable pattern. So this was kind of a way that the enzyme doesn't randomly chop up the bacteria's DNA, since the enzyme is specific for a sequence that the bacteria doesn't have in its DNA. So this way the enzyme only chops up the virus that's infecting it, not the bacterial DNA. So the picture shows a sequence of DNA that has been cut by a restriction enzyme called ecology. And this is because it comes from E. coli. So the enzyme recognizes the sequence GAATTC, and so it cuts after the G in the 5' to 3' direction on each strand. And the sequences that the enzyme recognizes are usually the same in both directions, similar to the one that you see in the picture. So many of the enzymes leave sticky ends on the fragments when they cut, and this allows the DNA sequences to be spliced into another vector if it's cut with the same restriction enzyme. And so therefore that other the other vector would have the same sticky ends that are complementary. So we'll go into more detail on how this works in a second. So the fragment of DNA that was cut out is then ligated into the new vector using the DNA ligase that seals the sticky ends back together. So if you combine the DNA of one organism into another, this is called recombinant DNA. So an example of this is splicing in the human gene for insulin into a bacterial genome. And this allows the bacteria to produce the protein in large quantities that diabetics use. So this way we produce insulin for a diabetic to inject to regulate their blood sugar. So since the human DNA sequences are not normally in that bacterial genome, this is a recombination of DNA, which gives the name recombinant DNA technology. So many proteins that are used in therapies and industry are now produced in this manner because bacteria are inexpensive to grow, and humans don't have reactions to the human proteins that are grown in bacteria. So in order to make recombinant DNA, a restriction enzyme is used to cut the DNA of the bacterial genome and also the donor DNA at a specific site, since the restriction enzyme recognizes a specific DNA sequence. So for instance, in the picture, the blue represents the bacterial genome, and the pink represents the gene that's being spliced. So for instance, say human insulin. So after the restriction enzymes cut both sets of DNA, the fragments have sticky ends. And so this means there's an overhang of base pairs that are not paired with another nucleotide. So these sticky ends, or this overhang, really wants to hybridize with matching nucleotides on another strand. So these fragments are then mixed together. So when the fragments are mixed, the matching sticky ends from the DNA base pair with each other, so DNA from the human gene and DNA from the bacteria are recombined in the same piece of DNA. The enzyme DNA ligase is then used to seal these pieces of DNA together. And the ligase repairs the nicks in the DNA backbone so that it is not just base pairing that holds the broken strands together. At this point, the molecules of recombinant or mixed DNA are a result. So the next thing we're going to look at is DNA cloning, and this uses recombinant DNA technology. So DNA cloning is a process used to mass produce a specific DNA fragment. So the fragment of DNA to be copied is inserted in the plasmid or other cloning vectors using the process for recombinant DNA that we just went over. And they're inserted in the host cells such as bacteria. So the host cells replicate the DNA when they divide, and they make identical copies or clones of the foreign DNA. The bacteria divide very quickly. So lots of the DNA you're interested in can be made very quickly in these bacteria. So this is essentially hijacking the bacteria cell's machinery, and you're able to trick it into doing the work for you. So the bacteria which contain the gene that was spliced in can also be used to express, meaning producing the protein. So they express large quantities of protein encoded by that gene if you want them to. So here are some terms that I want you to know. So DNA cloning is a set of procedures that uses living cells to make many identical copies of a DNA fragment. So this process usually is done by inserting foreign DNA into bacteria and then letting the bacteria divide like they normally do, which replicates the inserted DNA at the same time as they replicate their own. So the next thing is a plasmid. And a plasmid is a small ring of extra chromosomal DNA. So this means outside of the normal bacterial chromosome. And this is found in many bacteria and archaeans. So it's replicated independently of the chromosome, but it's always replicated and passed on to the daughter cells when the cell divides. So the picture shows how the plasmid is an extra ring of DNA separate from that bacterial genome. So these plasmids are one way in which bacteria transfer new traits, such as antibiotic resistance to each other. So a cloning vector is a DNA molecule that can accept foreign DNA. This is then transferred to a host cell and it gets replicated in it. So it's usually, a cloning vector is usually a plasmid that's been manufactured with specific sites where restriction enzymes will cut, and it also has specific markers such as antibiotic resistance or a tracer that allows the researcher to identify bacterial colonies that have taken up that vector. So this slide shows what a cloning vector map looks like. So on the left is a micrograph of an actual plasmid, and on the right is the map that tells the researcher which genes are in the vector and the location. So it also shows which restriction sites are present in the vector and where they are. So the order of the restriction sites is useful, so you can splice in the DNA in the correct orientation so that it's transcribed correctly. So generally, if you're transcribing in this direction, which is the way the arrows point, you would use, for instance, maybe a restriction enzyme cut there and one there so that you could orient the gene so that it's going that direction. And so you can see the size of the cloning vector. So this means it's 3,850 bases, so 3.85 kilobases. So that's quite a few nucleotides that are in that ring. And usually these restriction enzymes are recognizing about a 6 to 8 base pair sequence, so not one that's not very long. And so as you can see, there's a lot of these restriction sites in a very small part of this cloning vector. So in this vector, the kanamycin and ampicillin are antibiotic resistant genes. And so this way the researcher can select for colonies that have taken up that plasmid, and they do this by growing the bacteria on the antibiotics. So only those bacteria that have taken up the vector are able to grow in the presence of the antibiotics because they're resistant if they have that vector. So the LAC-Z gene is the lac operon that we learned about a couple weeks ago. And so this allows you to turn on or off the expression of the gene that you splice in. So the protein you're trying to produce would only be expressed in the presence of lactose, otherwise the gene is repressed. So researchers who study eukaryotic genes and their expression work with messenger RNA transcripts of the genes. So the reason why they use messenger RNA instead of the DNA in the genome is that messenger RNA has already had the intron spliced out. So remember that in eukaryotic cells, that the messenger RNA is processed before it's translated into a protein. And so therefore you can't just look at the genomic DNA because there's all those introns interspersed. So therefore, looking at the messenger RNA, this contains only the coding sections of the genes because the introns have already been spliced out. So RNA can't be cloned into a vector directly because it's single-stranded. So therefore, reverse transcriptase, this is a viral enzyme found in HIV and other viruses. This is used to transcribe a single-stranded RNA into the complementary DNA, or what's called cDNA for cloning. So in the first step, a strand of cDNA is assembled on the messenger RNA template by hybridization. And this allows the complementary base pairs to attach to the messenger RNA strand. Then DNA polymerase removes the RNA and copies the cDNA into a second strand of DNA. And so this results in a double-stranded DNA copy of the original messenger RNA. And so this now can be cloned into a cloning vector, which uses restriction sites such as the one highlighted, and then it's inserted into bacteria. So again, this is the ecol-R1 restriction site. And as you can see, if you read the top going this way, it's GAATTC. Reading the bottom going this way, it's GAATTC. And most restriction enzymes recognize sites that are like this, that are the same going both directions. So in order to isolate a specific gene from the entire genome, researchers have come up with ways to break the enormous genome into smaller bits that are easier to work with. So since bacteria are much, much smaller than eukaryotic cells, researchers also have to decrease the size of the segments they're working with because the DNA from a eukaryotic cell will not physically fit inside of a bacterium. So in order to do this, researchers randomly chop up genomic DNA or reverse transcribe all the messenger RNA in the cell and then clone all the fragments into a set of bacteria to create a DNA library. So a genomic library collectively contains all the DNA in the genome, whereas the cDNA library contains only those genes being expressed at the time that the messenger RNA were harvested. So DNA libraries, in a process that we'll get to in a second, allow researchers to sort through the huge amount of DNA in a genome or RNA from messenger RNA transcripts and isolate the particular DNA segment or gene they're trying to study. So this slide shows the process of how researchers use DNA libraries to isolate a particular gene or DNA fragment through nucleic acid hybridization. So the plate at the top shows a huge number of colonies grown on a bacterial plate. So each one of these colonies carries a different cDNA or genome fragment. And usually there are so many colonies that more than one plate is used. So a piece of special paper is then pressed on the plate and it picks up a little bit of the bacteria from each colony. The paper is then soaked in a solution that ruptures the bacterial cells and releases the DNA inside the cell, which binds to the paper in that same spot. So a probe is then used to identify a specific cDNA or genome fragment, and the probe hybridizes to the target DNA by base pairing to the complementary base sequence. So the probe also has some sort of label, such as a fluorescence or radioactivity, so that the spot on the paper it hybridized to can be identified. So the spot on the paper is then used to map out the colony on the original plate that contains a specific genomic fragment or DNA that you're interested in. So that way you're able to find that bacterial colony that has the DNA that you're interested in. So another technique that's used in microbiology that's used all the time is called PCR. So this is a polymerase chain reaction, and it uses primers and heat-resistant DNA polymerase to mass produce a particular section of DNA without having to clone it in living cells. So this process can amplify a couple of copies of DNA in the billions of copies within a few hours. So instead of cloning in the fragment of DNA in a cloning vector and growing it in bacteria, which is a process that can take a couple days to grow and isolate the DNA, if all you're looking to do is to amplify a specific sequence of DNA, PCR is going to be much, much faster. So PCR can be used on any sample of DNA with at least one molecule of a target sequence. So the process is very specific, and it can pick a needle out of a haystack when it comes to amplifying a single DNA sequence in the middle of millions of other nucleotides. So essentially any sample containing DNA can be used. So for instance, one sperm, a hair left at a crime scene, or a mummy as long as the DNA is not damaged. So a PCR machine cycles the reaction between high and low temperatures, and in each cycle of the PCR reaction, the target sequence of DNA doubles. So for instance, 30 cycles, which is the normal number of cycles of a PCR reaction, can make a billion copies of a single section of DNA. So this shows the process of a PCR reaction. So the DNA template, which contains the targeted DNA section, is mixed with primers. And these are nucleotides, so they're also mixed with nucleotides and a heat-tolerant DNA polymerase called Taq polymerase. So when the PCR reaction was first invented, polymerase would have to be re-added after each cycle because of high temperatures. So the high temperature in a PCR reaction is about 100 degrees Celsius, and this denatures most enzymes. So researchers then isolated the DNA polymerase from bacteria that grows in a thermal vent. So this bacteria is specialized to thrive and grow at very high temperatures. So therefore, the enzyme didn't denature at the high temperatures during each PCR cycle. So this Taq polymerase revolutionized the procedure because you can now add all the ingredients, start the machine, and then come back to billions of copies of the DNA sequence after only a few hours, instead of having to re-add that enzyme every cycle. So the primers in the mix are short segments of DNA that are designed to be complementary to each strand of DNA at the ends of the targeted section. So keep in mind, as we're going through, this highlighted portion right here is the segment of DNA that you're interested in and want to amplify. So you want more copies of that, only that section of the DNA. And in order to do this, you design primers that hybridize, or they're complementary to the sequence of this segment of DNA and this segment of DNA. So just the ends of the sequence that you're interested in. So when the mixture is heated, the double-stranded DNA template separates from the single strands because the heat is enough energy to break the hydrogen bonds holding the strands of DNA together. The reaction is then quickly cooled, and the primer's base pair are hybridized with the template DNA. So Taq polymerase then begins DNA synthesis at the primers. So complementary DNA strands form on the single-stranded template. So the process is then repeated for a second cycle, and the mixture is heated again. The double-stranded DNA separates into the single strands. The strands formed in the last cycle are now templates in the new cycle. And when the reaction is cooled again, primers base pair with both old and new strands of DNA. So each round of PCR reactions doubles the number of copies of the targeted DNA section because the new strands that are built serve as templates in the next round of amplification. So this process causes only the section of DNA that's between the two primers to be amplified to very large amounts. So there's going to be other DNA fragments in the reaction, but the ratio of the targeted sequence that you're amplifying to the other DNA is going to be very minute. So DNA sequencing is a process that reveals the order of the bases in the DNA. So the entire genomes of several organisms have now been sequenced, and this includes the human genome. So in the process of DNA sequencing, the DNA polymerase partially replicates a DNA template, and it produces a mixture of DNA fragments of different lengths. So these fragments are then separated by a process called electrophoresis to determine the sequence of the original template. So in order to understand how DNA sequencing works, you have to understand a little bit more about electrophoresis. So electrophoresis is a process that separates the fragments by length in the bands. So this can either be done done with protein or DNA fragments. So in the example shown in the picture, an electric field pulls the DNA fragments through a semi-solid gel. So the gel is usually made out of agarose, and this makes up a framework that has tiny holes through which the DNA travels through. So the DNA moves from the negative pole towards the positive pole of the box because DNA is a polar molecule with a negative charge. So therefore, it's attracted to the positive electric charge. So if you look on this box, you have a red electrode and a black electrode. So the red is plus, the black is negative, and so therefore the DNA is traveling in this picture from right to left. Because it's a negative molecule, and so therefore it's attracted to the positive pole. So fragments of different sizes move at a different rate. So therefore, shorter fragments move through the gel faster than longer fragments do. And so this means that the shorter a fragment is, the faster it will move down the gel. So the lane to the left is what's called a ladder. So I'm talking about this picture here. So this lane on the very left is called a ladder. It contains a series of DNA fragments that have known sizes. So therefore, we can compare the unknowns to it and know what the sizes are. So in the picture, the bands travel from the top to the bottom of the gel. So we're talking about in this picture right here now. So the bands at the bottom are DNA fragments that are smaller than the DNA fragments in the bands at the top of the gel. So therefore, it means this band right here is smaller than the one right above it, which is the longer one, because it moves through the gel faster. So the sequencing reaction depends on what's called dideoxynucleotides, to terminate DNA replication. So this nucleotide, dideoxynucleotide, has a hydrogen at the 3' carbon of the ribose instead of a hydroxide. So therefore, it can't bind to the next nucleotide. So each of the 4 dideoxynucleotides is labeled with a different colored pigment. So in the slide, adenine is green, cytosine is blue, guanine is black, and thymine is red. So the DNA that's supposed to be sequenced serves as a template to build a new strand of DNA. And the dideoxynucleotides are mixed with unlabeled deoxynucleotides and serve as the monomers to build the new strand. So the dideoxynucleotides that are labeled, these terminate the reaction versus the unlabeled deoxynucleotides, which are used to build a new strand. So as the new strand is being built, the dideoxynucleotides, which are in a much smaller ratio, are going to be randomly inserted into the DNA sequence instead of the normal nucleotides. And whenever this happens, it automatically stops the replication. So the template DNA is copied many times, and at the end of the sequencing reaction, the mixture contains many incomplete copies of the original DNA. And the length of those copies depends on how long it took for the reaction to randomly insert a dideoxynucleotide instead of a regular one. So electrophoresis then separates out those DNA fragments into bands according to the length. So all the DNA strands in each band are the same length and end with the same dideoxynucleotide labeled with the same color. And so you can see how this works. A computer detects and records the color of each successive band on the gel, and the order of the colors represents the sequence of the template DNA. So depending on what color a band is, determines which nucleotide is at that position. So the readout at the bottom is what the results of the sequencing reaction look like. So the peak of each color tells what in the nucleotide, what nucleotide base it is. So the human genome consists of about 3 billion bases. So starting in the 1980s and accelerating in the 1990s, many groups set on the task of sequencing the human genome. So the idea was that the more we know about the genes and how the other DNA is used to regulate the expression of the genes, the more power we would have to fix it when things go wrong. So hoping to patent the sequence, a company called Cellular Genomics invented faster methods of sequencing genomic DNA using supercomputers. And so by 2003, the human genome sequence was completed. But governments, including the US and Britain, decided that the sequence of DNA in the human genome was not something that could be patented. So unfortunately for that company, they didn't get the benefit of being the first to sequence. So the picture on the left shows some of the supercomputers used to sequence the human genome at Cellular Genomics. And the picture on the right shows the raw data of a human genome, a human DNA sequence. And so you can see on the right just how complicated that sequence is. So again, we have 30,000 known genes. And those are the actual genes that are translated into proteins. So therefore, there's all the regulatory DNA, and then also the introns that don't have anything to do. And that we're still actually learning the purpose of a lot of those introns. So you can imagine just how many bases are involved in the human genome. So genomics is the study of genomes, and it provides insights into human genome function. So it includes whole genome comparisons, structural analysis of gene products, and the study of small-scale variation among individuals. So even though full genome sequencing is available now, it's going to be a long time before we understand all that coded information. So initially, when the Human Genome Projects were announced, it was thought that once we know the full sequence of DNA, that somehow it would open up a magical box where we would suddenly understand so much more about our genome and be able to cure genetic diseases. So sifting through the 3 billion bases and understanding the meaning of it has turned out to be a lot harder than originally thought. So there have been many breakthroughs from having the entire genome sequenced, but we've only touched the tip of the iceberg of what there is to come, of figuring out what it all means. So not just the human genome has been sequenced. So the genomes of many animals have also been sequenced, and this allows comparisons to be made. So comparing genomes of different organisms shows evidence of evolutionary relationships, and it's also used in medical research. So the picture shows a region of the gene for DNA polymerase where the difference is highlighted. So the chance that any two of these sequences would randomly match is about 1 in 10 to the 46. So the sequences for humans, mice, rats, dogs, and chickens are exactly identical. And the DNA polymerase in the other organisms listed don't vary by much either. And that includes frogs, zebrafish, and fugu. So the similarities between the DNA sequences between all of these organisms shows that they're descended from a common ancestor. So DNA profiling is a process that identifies an individual by analyzing the unique parts of his or her DNA. So this process can be used for paternity tests or criminal investigations. So examples of DNA profiling include determining an individual's array of short nucleotide polymorphisms, or SNPs, on microscopic arrays, and also analysis of short tandem repeats. So short tandem repeats are sequences of 4 or 5 bases repeated multiple times in a row, and they're found in chromosomal DNA using PCR. So this slide shows an individual's short tandem repeat profile. So each peak on the chart represents a test for one short tandem repeat. So as you can see, there are 2 peaks for each locus because you have 2 homologous chromosomes that have the same locus. So the size of a peak indicates the number of repeats at that locus. So for example, this individual has 14 repeats. That's a D3S1358. So this is that first one right here. In chromosome 3. They also have 17 repeats on the homologous chromosome. So you can see there's 14 and 17. So for the D18S51 locus, this is the 4th locus right here. They have 14 repeats on one chromosome and 18 repeats on the other one. So the number of repeats changes through generations. It can go up and down. And so nobody besides identical twins have the exact same number of repeats at more than a couple of loci. So testing is usually done looking at 10 to 15 loci, and this gives a completely unique profile for each person's DNA. So that's what people refer to as your DNA fingerprint. So genetic engineering is a process by which deliberate changes are introduced into an individual's genome, producing a genetically modified organism, or GMO. So a gene may be altered and reinserted into an individual of the same species, or a gene from one species can be transferred to another to produce an organism that's transgenic. So the most common GMOs are bacteria and yeast, like bacteria, that have been modified to produce medically important proteins such as insulin. So engineered bacteria also produce enzymes used in food manufacturing. So this includes chymotrypsin, which is used to make cheese. The proteins also these proteins used to be extracted from animals. So for instance, we used to extract insulin. This was a much more expensive process, and also animal proteins can cause allergic reactions. So the picture on the right shows E. coli modified to produce a green fluorescent from a jellyfish. So the picture on the left shows all the fluorescent colors that have been cloned in the bacteria from mutations in that green fluorescent protein. So the beach scene was drawn in a petri dish using different colonies of fluorescing bacteria. And so this is just actually done just for fun. These different colors that are used in these fluorescent proteins are extremely important in scientific research to trace various proteins. So you can connect this fluorescent protein on whatever protein you're interested in studying and locate it within a cell where that protein is localized. So it's extremely important in scientific research. So genetically engineered crop plants are widespread in the U.S. And researchers use a tumor-inducing plasmid from bacteria as a vector to transfer foreign or modified genes into food crop plants, such as soybeans, squash, and potatoes. So transgenic crop plants can be resistant to diseases. They offer improved yields or make a protein that's toxic to insect larvae. This allows less insecticides to be used. So the bottom pictures show the process of creating a genetically engineered plant. So the TI plasmid that contains the gene you would like to add to your plant is inserted into a bacteria. And the bacteria infects the plant and transfers a plasmid into it. The plasmid DNA becomes integrated in the plant's genome, and the plant cell then divides, forming an embryo that then grows into a plant. So the picture on the right shows a plant that contains the luciferase gene. So this was a gene from the firefly that makes it glow. So I'm not sure what the point of making a tobacco plant glow, but I think this was just to show that they can do it. So this slide shows how inserting the BT gene into corn makes the genetically modified plants insect resistant without using chemicals, as you can see in the top picture. So the insect larvae eat the corn that contains the BT gene and are immediately killed so they can't continue to eat the crop. So corn produced by unmodified plants is more vulnerable to insect pests because the larvae grow up and lay more larvae, and the crop becomes destroyed like the picture you see on the bottom. So transgenic crop plants engineered for drought tolerance and nutrition are being developed for impoverished areas. So in countries in Africa where there are consistent droughts, this could help millions of people eat. Also, vitamins can be added, such as rice plants engineered to make beta-carotene, which is a precursor of vitamin A in the grain. So more than 2 million children die every year from vitamin A deficiency, and this crop would help alleviate that. So the most widely planted GMO crops include corn, sorghum, cotton, soy, canola, and alfalfa, engineered for resistance to glyphosate, which is an herbicide. This is the same chemical that you use that you see in Roundup. So unfortunately, GMO use is a controversial issue. They've been banned in Europe. So the crops are not tightly regulated and they can contaminate the environment and other crops. So the engineered gene, for instance, for glyphosate, resistance is occurring in wild plants and in non-engineered crops. So this means that transgenes can and do escape into the environment. So many people are also opposed to any GMO crops. They call such foods frankenfoods. But humans have been altering the genetics of plants for thousands of years through crossbreeding. And so selecting beneficial traits in that process, people generally don't have a problem with. But the idea of genetically modifying or constructing a new plant by messing with the genes generally is too much for a lot of people. So there's also questions now about other diseases being caused by GMO plants. I haven't really seen anything that's concrete, but as with everything, people jump on it, and there's now a big push against using the GMO crops. So genetically modified animals are invaluable for medical research and other applications. So genetically modified mice are used in gene research, and they're used as models of human diseases. So for instance, in my research, I worked with mice who had a defective ApoE gene. And so therefore they couldn't transport and metabolize cholesterol and lipoproteins correctly. And so they, in the end, would develop heart disease. So without these mice, research would have to be done on humans, and medicine therefore would be much further back in the treatment of the disease. So GM animals also make proteins that can treat cystic fibrosis, heart attacks, blood clotting disorders, and nerve gas exposure. GM goats produce spider silk protein in their milk. This allows silk to be produced much more cheaply and in larger quantities. GM rabbits make human interleukin-2, and this helps the immune system to fight off diseases. So, also GM farm animals can produce higher yields of meat and milk. So these pictures show some examples of genetically modified animals that are used in research and medicine. So the picture on the left is a transgenic animal that had the yellow fluorescent protein spliced in. And it shows on the right the non-transgenic littermate. So Mira, the transgenic goat, produces a human anti-clotting factor in her milk, and this is used for the treatment of hemophilia, which is the blood clotting disorder. This slide shows one example of how genetically engineered animals are useful in research. So mice transgenic for multiple pigments, called rainbow mice, allow researchers to map the complex neural circuitry of the brain. So individual nerve cells in the brainstem of rainbow mouse are visible in the fluorescence micrograph. So the fluorescence that labels each cell allows a researcher to visualize the nerve cells track through the brain. So one idea for the use of genetically modified animals is to use them to grow human tissues for transplantation. So scientists are a long way from being able to grow a transplantable organ in a petri dish. Human donors for organ transplants are in short supply, and donated organs are subject to rejection. Genetically modified animals may one day provide compatible organs and tissues for what's called xenotransplantation into humans. So animals can be modified with DNA from the recipient to grow an organ that's an exact match with no chance of rejection. So unfortunately, researchers don't know whether new technologies might produce super pathogens or new dangerous forms of life. So some of the worst pandemics have been caused when viruses cross over and become infectious to humans from animals. So this is one of the main concerns with the bird flu a couple of years back, and also with the fear of the H1N1 virus that came from swine back in 2009. So safety guidelines written by government agencies minimize potential risk to researchers in genetic engineering labs, and government regulations limit release of genetically modified organisms into the environment. But none of these are guarantees against accidental releases or unforeseen environmental effects. This is something, unfortunately, where there isn't a perfect answer. So the opportunity to cure diseases and increase crop yields to feed more people is in front of us, but many people don't think the benefits are worth the risk, and that they believe that the risks are too great. So scientists are also looking for ways that they can correct the genetic disorders that people are born with. So with gene therapy, a gene is transferred into a body cell to correct a genetic defect or treat a disease. So as with any new technology, potential benefits of genetically modifying humans have to be weighed against the potential risks. So when we as a society continue to work through these ethical implications of applying new DNA technologies. So gene therapy is now being tested as a treatment for heart attacks, sickle cell anemia, cystic fibrosis, hemophilia A, Parkinson's, and Alzheimer's diseases. Also several cancers, inherited diseases of the eye, ear, and also the immune system. So one disease that scientists tried to treat with gene therapy with mixed results was called SCID-X1. So this is severe X-linked disorder of the IL-2RG gene, and this codes for an immune system receptor protein. So affected children can't fight infections, and they only survive in germ-free isolation nets. And these kids used to be called bubble boys because they could not come out of the plastic isolation bubble without risk of acquiring an infection they wouldn't be able to fight off. So in the 1990s, 20 boys with SCID-X1 were treated with gene therapy. So researchers used a genetically engineered virus to insert unmutated copies of IL-2RG in the cells taken from the bone marrow. So 18 of the boys were cured. The boy in the photo, Rhys Evans, was born with SCID-X1, but his immune system has been permanently repaired by the gene therapy that was performed. So unfortunately, the procedure did not go perfectly because no one could predict where the virus-infected gene will insert itself into a chromosome. So if the virus inserts itself in the wrong place, it can cause many problems, such as if it interrupts a gene that controls cell division. This can end up leading to cancer. So 5 of the 20 boys treated with the gene therapy for SCID-X1 developed bone marrow cancer leukemia, and 1 ended up dying. So in another experiment, a young man with another genetic disorder had a severe allergic reaction to the viral vector, and 4 days after the gene therapy treatment, his organs shut down and he died. So the gene therapy experiments were first done on kids with disorders like this because their quality of life without the treatment was so poor. But someone with diabetes or other genetic diseases that have less impact on their quality of life, in those situations the risk wouldn't be worth it. So there's still a long way to go with these gene therapies, and hopefully at some point, even with these diseases that don't have as much of an impact, hopefully someday we'll be able to cure type 1 diabetes and these other genetic diseases. So using various methods to select the most desirable human traits also raises ethical issues. So it sounds reasonable enough to make sure that your baby is free from genetic diseases, but is it acceptable to genetically engineer cuter babies or superhumans with greater strength or intelligence? What about preventing obesity, aggressiveness, or homosexuality? So these are not simple questions to answer since everybody has different views on what's important. So this is also a sensitive issue because eugenics, or the idea of deliberately improving the genetic qualities of the human race, brings back memories of the Holocaust and the experiments that were done in the concentration camps. So thankfully we're not at the point where it's possible to choose any of these things, but someday we might be, and it would be a good idea to have rules in place before that time. So scientists usually attempt to determine whether we can do something, not whether or not we should. So that's the end of this chapter. So again, as a reminder, the test will be this Thursday at 8 o'clock. Good luck studying. I posted the answers to the review questions, and just let me know if you have any questions while you're studying. Have a good night.